Good afternoon, students. Today we are going to discuss about qualitative analysis of proteins. Here we are going to study various tests that are used for identification of protein present in the sample. Okay, so as a routine practice, first when we are given a sample, we have to perform Molish test and check whether it is a carbohydrate or not. If we come to know it is a non-carbohydrate compound, then we go for whether it is a lipid or a protein. If the compound is protein, it gives biuret test positive and urea nitrate test negative. Okay, so let us continue for the test for proteins. What are proteins? They are complex specialized molecules. They are made up of carbon, hydrogen, oxygen and nitrogen. Many proteins also contain sulfur in them, and amino acids are the building blocks of proteins. So let us now directly discuss about what are the tests that are used for detection of proteins. Okay, we are going to study 6 different tests. First is Biuret test, Ninhydrine test, xanthoprotic test, Valence test, Nitro procide test and heat precipitation test for the detection of proteins. So let us first discuss about Biuret test. Biuret test of proteins positively identifies the presence of protein in a solution. And if protein is present in the sample, we get a deep violet color. Actually, biuret is a compound which is formed by union of two urea molecules. So any sample which contains urea also gives this test positive. Okay, so biuret, upon heating, proteins form a compound biuret because they also contain amide linkage. The biuret compound reacts with copper sulfate under alkaline condition. Okay, biuret reacts with copper sulfate under alkaline condition and forms a deep violet color. If protein is absent, the color will remain blue. But formation of deep violet color indicates that protein is present in the sample because a compound called as biuret is formed. Okay, so this is the structure of a compound that is called as biuret, where there is union of two protein molecules and it reacts with alkaline copper sulfate solution to form deep violet color. Okay, so take 2 ml of protein solution in a test tube and add few drops of say 10% of sodium hydroxide solution, and then you add to it few drops of 0.5% copper sulfate solution and wait for the formation of purple color, deep violet color or purple color. Formation of purple or deep violet color as shown in this diagram indicates that the compound is protein. So the next test is Ninhydrine test. Ninhydrine test is especially used for identification of amino acids and proteins. Okay, also to differentiate proteins from carbohydrates. Now Ninhydrine combines with amino acids that are present in protein. Okay, two molecules of Ninhydrine combine with one molecule of amino acid and forms a purple colored complex called as Ruhemann's purple. Okay, almost all amino acids give purple color except proline and hydroxyproline, which forms yellow colored complex. So Ninhydrine is a compound which is commonly used to identify proteins or amino acids, and this test especially is used in forensic laboratory to detect fingerprints, because even fingerprints contain proteins and hence they give purple color. Okay, so take 1 ml of amino acid solution in a test tube and add 2 to 3 drops of Ninhydrine reagent to it. Wait for some time or heat, put it in a boiling water bath and check the development of purple color. So if purple color is formed, the compound is protein or the compound contains amino acids. The only difference is proline hydroxyproline will give yellow color instead of purple color. So 1 ml of protein is to be taken in a test tube as shown in the diagram. You have to add 5 drops of Ninhydrine. Either allow it to react in cold or slightly heat it by keeping the tube in a water bath and check for the development of purple color. So development of purple color, that is formation of purple colored Ruhemann's complex, indicates positive test. The next one is xanthoprotic test. Xanthoprotic test is specially given by amino acids that contain activated benzene ring, or you can say which are aromatic amino acids. Okay, so xanthoprotic test is done for checking the presence of aromatic amino acids in proteins. So some of the amino acids like tyrosine, tryptophan, phenylalanine contain aromatic rings. Okay, but only tyrosine and tryptophan contain activated benzene ring, and hence proteins which contain tyrosine or tryptophan give this test positive, because this is a test of activated benzene ring. Okay, nitration of benzene ring takes place and development of red color forms. Okay, phenylalanine doesn't give this test positive because it does not contain activated benzene ring. So you have to take 2 ml of protein solution in a test tube and add 1 ml of concentrated nitric acid and heat the solution for 2 minutes and cool it. When you cool it, yellow color complex is formed. Formation of yellow color complex indicates the presence of aromatic amino acids, okay, and hence that the compound is a protein. And if you add few drops of 40% NaOH, then orange color will be formed. So development of orange color after addition of few drops of 40% NaOH indicates positive xanthoprotic test and hence presence of proteins in the given sample. The next test is Millen's test. Millen's test is a specific test for tyrosine. Okay, the only amino acid which contain phenolic OH group in it. Okay, phenolic OH group attached to benzene ring. So Millen's test is given positive by proteins that contain tyrosine amino acid in it. Okay, in Millen's test, the phenol group of tyrosine is first nitrated by adding nitric acid, and then it reacts with mercury to form a red colored complex, that is red in color, or red colored precipitate is obtained. Okay, so Millen's reagent is added to a protein, okay, and a white precipitation is formed which turns brick red upon heating after adding Millen's reagent. So you have to take protein solution, add Millen's reagent to it and heat it. If red colored complex or red color precipitate is formed, then the compound is a protein because it contains amino acid tyrosine. Right. The next test is Nitro procide test. Nitro procide test is specifically done for to check the presence of amino acid containing sulfur group, sulfhydryl group, SH group. Okay. So proteins that contain sulfhydryl group containing amino acids like cysteine or cysteine will give this test positive, because this is a test for detection of sulfhydryl group. This group, that is sulfhydryl group, reacts with nitroprusside, sodium nitroprusside, in presence of excess of ammonia. Okay, so you have to take 2 ml of protein or amino acid solution in a test tube. To it you add 0.5 ml of sodium nitroprusside and then shake it, and then add 0.5 ml of ammonium hydroxide. Okay, so 2 ml protein or amino acid solution, 0.5 ml sodium nitroprusside and 0.5 ml of ammonium hydroxide solution. Okay, so sodium nitroprusside reacts with the compounds that contain sulfhydryl group, that is proteins, and produce a intense red color, but the red color is quite unstable. Okay, so you have to take the sample in a test tube and add few drops of sodium nitroprusside and then ammonium hydroxide, and you can get a deep red color as shown in the figure, deep red purple color as shown in this figure. So presence of this color or appearance of this color indicates positive test for proteins. Okay, the last and most important test which is also used for urine analysis is heat and acid precipitation test, or also called as heat and acid coagulation test. Okay, so albumin that is present in the sample, this test is specially given by protein albumin. So if you heat the sample, okay, in this case we have to fill 3/4 of the tube with the sample and heat the upper portion of the tube. When you heat the sample directly on heat, the proteins present, especially albumin, will get precipitated upon heating. So the upper part of the tube where you have heated the protein will show precipitation, whereas the lower part which serves as control will be clear. There won't be any appearance of turbidity. Okay. Now to confirm that this turbidity is formed only by protein, the turbidity is not because of precipitation of carbonates, it is not because of precipitation of phosphates. That is, the turbidity, appearance of turbidity is not because of protein only. That means it is not a false positive test. To confirm that, we have to add few drops of glacial acetic acids to the test tube. After addition of glacial acetic acid, if the turbidity or precipitation persists as it is, means the precipitation is because of proteins and that proteins are present in the sample. If the precipitation or coagulation disappears upon addition of acid, it indicates that the test is negative. The precipitation was false. It was not because of protein, it was because of carbonates or phosphates. Okay. So, heat precipitation test is to be confirmed by addition of acid. So, that is all about identification of proteins by various tests.